And the next presentation will be by Gema Fuerte Hortigon from Mission Bio, Spain. Gema will present the results about the data information about Mission Bio's Tapestry platform, multi-omic SNV, CNV, and protein sequencing from thousands of single cells. Gema, please.
Yes, thank you. Thanks a lot for inviting me. I hope you can see my screen now. Yes. So yeah, my name is Gema Fuerte, and I am the European Field Application Scientist at Mission Bio. So this is the agenda of what I will be showing you today. So the first one will be why single cell is important. Also an overview of the Tapestry platform, which is the platform we have here at Mission Bio. And then I'll show you the two latest publications on multiomics, DNA plus protein. One has been published in Nature and the other one has been published in Nature Communications. So for those who don't know what Mission Bio does, so this is just an example and a slide that I like to use a lot about the importance of heterogeneity. So we know that cancer is quite heterogeneous and often there are different populations within a cancer mass. These populations might be defined by somatic mutations, by copy number changes, and they might be different cell subtypes based on protein expression of membrane markers. And here I know I'm going to be talking about cancer all the time, but please think about this as an heterogeneous tissue as well. It does not need to be cancer. So what Mission Bio platform can do is can decipher the exact heterogeneity of a tissue or a cancer sample or a blood sample. So for example, if this is a leukemia, we see different subclones here defined by different somatic mutations. We can see exactly what somatic mutations we have, the co-occurrence of the mutations in the same or different populations, and also the zygosity. So we are a single cell DNA sequencing platform, and we are the only ones that provide somatic mutation information, and also now copy number and now protein plus DNA at a single cell level. So how do we achieve this resolution? So unlike bulk next generation sequencing, which is a population-based analysis, and when you do bulk, you have millions of cells, you sequence that, gets millions of reads, and then get a VLEL frequency averages of the mutations found in the sample. Like for instance, here we have IDH1 and IDH2 represented in our sample. With the Tapestry platform, what we do is we barcode each and every single cell up front the sequencing technique in a way that then we can tell you which mutation is found in which cells. So as you can see here, IDH1 and IDH2, there is no issue usually to detect these mutations with bulk because they are present in majority of the cells, right? In high percentage of the cells. But then the challenge starts when you are interested to determine somatic mutations that are present in small populations in 0.1, 2% or 1% of the cells. So with the Tapestry, you can certainly do that at the same time as you identify the co-occurrence with other somatic mutations present in that subclone. And that's the data I'm going to be showing you today. So why is how is single cell DNA used in the context of cancer research? So this is a very nice review from Dr. Nick Navins and MD Anderson. And they go through different single cell techniques. They go through RNA-seq, ATAC-seq, and also DNA-seq with the Tapestry platform. So this is what they use the DNA-seq single cell for. So as you have very good resolution, of course, at the single cell, as I said, you can determine the exact clonal substructure of your samples. You can also determine phylogenetic trees, so which mutations are found in your cancer samples or in your patients in the order of these mutations. When are these mutations acquired? You could also follow a tumor evolution. So what clones are present, like let's say in a context of minimal residual disease, when you have one patient with diagnosis, clinical remission, and relapse, what clones are in the diagnosis, what clones are in the relapse. So you can do that. And as well as I said, the co-occurrence of the mutations and the zygosity. So whether the two mutations are in the same population and heterozygous or homozygous. Because 50% VLEL frequency can mean all the cells are heterozygous or 50% of the cells are homozygous. So this is something that up until now was inferred from bulk sequencing, but we can provide the real information at a single cell level. So with the Tapestry platform, you can do that and many other things. So as I said, now we are a multi-omics platform. So we are the only platform out there that can provide you single cell DNA and protein profiling. When I say DNA, I mean somatic mutations, insertion deletions, and also copy number changes paired with protein expression markers across hundreds of targeted loci. So we are a targeted platform and we work with amplicons. Our panels are amplicon based. So you could essentially create a custom panel anywhere between 20 to 1,000 amplicons. The copy number variation information that we provide is a gene level. It can also be exon level. It can even be chromosome level. We could just design amplicons against the entire chromosome and provide you the information of copy number. In terms of throughput, we are a high throughput platform. So our throughput is 5,000 to 10,000 cells, starting with 100,000 cells. So it is a 5 to 10% capture rate. The sensitivity is also very, very high. So we can go down to 0.1% when working with individual samples, so individual time points. But if you are working with multiple time points, such as in the context of minimal residual disease, if you determine one pathogenic mutation is important either at the time of diagnosis or at the time of relapse, it will show up in the other time points, such as the clinical remission, even if that represents one cell. And that could be 0.02%, for example. In terms of the content of our panels, we have some catalog panels for specific cancer indications. I'll show you a screenshot of that. And we also do custom panels. So this means, if you are working in a specific indication, which doesn't need to be cancer, as I said at the beginning, we can just create amplicons against your regions of interest. In terms of tissue types, we have worked with many, many different tissue types. So the first advantage is we can work with fresh and frozen. For hematological malignancies, we can work with PBMCs, we can work with bone marrow, of course, with cell lines. We can even work with alcohol fixed samples. For solid tissue, we can work with whole cells, but also isolated nuclei, which is a great advantage as well for perhaps old frozen solid tissue samples. And what we have yet not validated is FFPE at the time of for the single nuclei process. But I will show you some, for those interested in solid tissue, I will show you some examples of cell throughput that we have achieved. With our platform, you can see here cell lines and PBMCs and bone marrows, a high cell throughput. As I said, it is anywhere between 5,000 to 10,000 cells. And then you have here another solid tissues. We have tested many different tissue types: colorectal, prostate, pancreatic, brain, glioblastoma. And this is the kind of throughput that we have obtained. So for those who wanted to work with isolated nuclei, we also have a protocol available on our website just to help people do the nuclei isolation in case they are not experienced, doing this sort of isolation. So in terms of the solution, Mission Bio offers an end-to-end solution. So we offer the reagents, you purchase the reagents, and you have everything you need to get from single cells to sequencing ready libraries. After we also, and as I said here, the content, as we will see later in a couple of slides, could be catalog or custom. We also provide the platform. It is this very easy to use benchtop equipment platform, very small, and to run disposable microfluidics cartridges. So you will be using one cartridge per sample, and the cartridge is really where everything happens. I'll show you the workflow later. And then we also provide the tools for analysis. So this is extremely valuable when working with single cell data because we have different software types. We have a pipeline to do the primary analysis. Then we have a GUI, very easy graphical user interface to do secondary and tertiary analysis. For those who don't have extensive bioinformatics background, is great because you can get to nice visualizations of your clones without having to do much work or have extensive knowledge on bioinformatics. And we have that included in the price of the reagents, which means you don't need to pay extra to analyze the data. So in terms of the workflow, it integrates very seamlessly with an existing NGS workflow. So Mission Bio is compatible with all Illumina sequencers, 2 times 150 base pairs. And as you can see here, you would be preparing your cells in suspension or your isolated nuclei in the lab and using the Tapestry platform to, let me just put the laser pointer. So prepare your cell suspension or isolated nuclei and use the Tapestry platform to get to the sequencing ready libraries. So from here to there, you only take 1.5 days. You could start on Monday morning and on Tuesday evening, you could be sequencing your single cells. Once you sequence them, as I said, with any Illumina platform, we are compatible with all of them. You get the raw data and then you will be using our software solutions to do the analysis. So what happens here? I'll take you through our workflow. So as I said, we work with disposable microfluidics cartridges. This is just a cartoon of what the cartridge would look like. So our workflow is unique. It is a unique 2-step encapsulation workflow. The first part here is called encapsulation, and it is when we encapsulate single cells in 40 micron oil droplets with a protease solution. This takes 5 minutes and we follow 5% Poisson distribution. So this is to avoid, this means that we generate millions of droplets and only 5% contains a cell. That is to avoid 2 cells being encapsulated together. So we minimize the number of doublets that you will have at the end. This process only takes 5 minutes. So after you do the encapsulation and you have single cells in suspension inside of droplets, we proceed to lysis. We put the cells on a thermocycler and then we digest all the DNA binding proteins, all the membrane proteins, and we have a nice genomic DNA lysate as for a single cell inside of a droplet. And then we proceed to the second step, which is the barcoding. Here is where we will be combining the single cell, the genomic DNA of a single cell, with all the primers needed and that unique barcode in order to amplify PCR, multiplex PCR overnight, to amplify these loci of your interest. So you will be combining the primers, the enzymes, your barcode, and your genomic DNA to do a multiplex PCR overnight. Essentially, what you generate is a nano PCR reactor where you have all the ingredients, if you wish, you require in order to do this amplification overnight. This is day 1, and day 2 is standard library preparation. So you break the emulsions and clean up the DNA and add the adapters for sequencing. Now, at the beginning, I said that now we don't only provide DNA, we also provide DNA plus protein simultaneously. So the way we achieve this is the workflow is the same I just described, but you have an additional step upstream of the protocol where you stain your cells with antibodies that are conjugated to a DNA oligo. So then, after you stain your cells with these antibodies and they're conjugated to a DNA tag, you pass them through the workflow. So at this point, you have your cells with all the antibodies bound to the surface, you encapsulate them, and at this point, you digest and you lyse. And of course, I mean, the same way as you lyse the protein, the membrane, you will also be lysing the antibodies. But the tag of the antibodies will remain. This is what it is depicted here with a green dot. This means that when you go through the barcoding protocol, the DNA and the protein tags will be amplified with the same barcode. So you will essentially generate 2 libraries per cell. You will generate a DNA library and you will generate a protein library, which is the tags of the antibodies that bound to the surface. And they both will be amplified with the same barcode. So then bioinformatically, you know, the cell with this and this mutation express this and this protein marker. And that's how we achieve the multi-omics approach. Okay, so let me show you some examples of some panels that we have available. For hematological malignancies, we have 3 catalog panels. We have an acute myeloid leukemia panel, a myeloid panel, and a chronic lymphocytic leukemia panel, plus some more pre-designed that I'll show you in the next slide. For solid tumors, we have 1 tumor hotspot panel targeting the most common oncogenes and tumor suppressor genes, and also this universal nuclei isolation protocol. So because we have very good resolution at a single cell level, we have also explored other techniques which are not cancer research, for example, gene editing or CRISPR-Cas. So you can imagine if you edit a population of cells, you can target these edits as if they were somatic mutations. So that's why with our custom panels, we can help with this type of applications as well. And we are compatible with human and mouse. This is just a larger list of other panels that we have available on our website in case you are interested in any specific indications that are listed here. As I said, if you are interested in a completely different application, we have an excellent option to do a custom panel. So we will just create whatever amplicons you require. All right, so after I've done the overview of the platform, I'll move on to showing you our two latest publications. So these are both in Multiomics DNA plus protein, which is the new data our customers are generating right now. The first one is a large single-cell DNA atlas of acute myeloid leukemia patients. As a background, this study is from Dr. Koichi Takahashi from MD Anderson Cancer Center, and this was published in Nature Communications last month. This is the largest atlas done on single-cell DNA sequencing for acute myeloid leukemia patients. They analyzed more than 500,000 cells from 77 different patients. And what they were interested here was to understand the heterogeneity of this disease, which of course it was known and analyzed before with bulk approaches, but this was the very first time this was shown at the single-cell DNA level. So they wanted to know the heterogeneity of these patients, also the pattern of these mutations, whether they are in the same, they tend to co-occur in the same population or happen in different populations. And they combined it with protein expression markers. So are my malignant clones, do my malignant clones belong to the same cell type, or they are perhaps different cell types? So the first thing I wanted to show you is a very big snapshot of the 500,000 cells and the 77 patients, where you can see all the mutations that were assessed from top to bottom and whether they were mutant or missing. So as you can see here, for example, there are some mutations that seems to be mutually exclusive. This means that when one happens in one clone, it does not co-occur with the others. This was a thought for, like, for instance, these IDH1 and IDH2 mutations, that they might probably be mutually exclusive. So here on the left, you can see as well the number of sequenced cells. So that speaks to the platform, to the high throughput of the platform. You see there is an average of 7,500 cells per sample. And the correlation with bulk sequencing is also really, really good. So this is a very powerful snapshot of all the cells together. You see here, this bucket corresponds to 10,000 cells. These are all the patients that were analyzed. So very nice to see at a single cell level, these co-occurrence or mutually exclusivity patterns. So then if you move more towards the evolution of these acute myeloid leukemia patients, you see they determined, or they described different patterns of clonal evolution. So here you have two patients where you can see clearly the acute myeloid leukemia samples underwent linear evolution. So one clone acquires one mutation, then another mutation and another mutation. Like these ones here that underwent branched evolutions, when one clone acquires one mutation, at the same time acquires another mutation and another mutation, so the phylogenetic tree becomes larger and more complex. They also described that the clones expected in the branched evolution samples are much higher, the number is much larger than the clones you expect in the linear evolution samples. But what does it mean? So, of course, there are different types of acute myeloid leukemia, and they were also interested. One, you now know the pattern of co-occurrence of mutual exclusivity. You also know the evolution, what mutations are acquired and in which order. And then we move on to the clinics of it. So what we see here are fish plots. For those who are not familiar with fish plots, this is a representation of different clones encountered at different time points. So you have here baseline, clinical remission and relapse, baseline, clinical remission and relapse of 2 patients here that were treated with chemotherapy. Achieved clinical remission for a while, all the clones subsided, and then unfortunately relapsed. And you can see the same here for this patient on the right. Okay, so what you can see here with these fish plots are all the clones that were encountered at the time of diagnosis. For instance, this one, FLT3835, is the clone or the mutation driving the relapse that was actually present at the time of the diagnostic. This has a huge importance when analyzing mechanisms of resistance, or even, you know, doing clinical trials. So deciding which patients you're going to include in a clinical trial, because you see a pathogenic variant that might expand in the future is already present at the time of diagnosis. So super, super interesting. You see as well, these 2 patients are less heterogeneous, and you don't have so many clones at the baseline, and often the clones at the relapse are different than the ones at the baseline. What we see here below are 2 fish plots of patients that unfortunately never achieved clinical remission. So as you can see, they were super heterogeneous, and they had many more clones at the beginning, at the baseline to start with. They were treated with chemotherapy, but also with targeted therapy against FLT3 mutations. You see the FLT3 clone go down, FLT3 single clone also go down, but the patients are so heterogeneous and they have so many clones that don't allow the patient to respond. So they never achieve clinical remission. Same we can see here. There is a big FLT3 ITD clone at this time of diagnosis that goes down because, as I said, they are being treated with targeted therapy, but there are so many clones in the background that just unfortunately don't allow this patient to respond. So they remain refractory. These fish plots has been done for a long time with bulk sequencing, believing that the mutation with a higher V allele frequency is the founder mutation, and then the others are subsequent mutations. But we see that's not often the case. When you analyze it at the truly single cell level, you see the true heterogeneity of your cancer samples. So if you want more information, this was published, as I said, in Nature Communications this year. And then to finish off with this specific study, they also did the multi-omics approach, right? So after they analyzed all of these patients and they determined what clones they found, they were interested to see if the cell type of the mutant of the clones encountered was the same or different. So this is just an example of a single patient that had one single TP53 clone, then a double TP53 clone, and also a triple TP53 double clone plus KRAS. So it is a linear evolution. You acquire 1 mutation, 2 mutations, and 3 mutations. So when you do the DNA plus protein simultaneous analysis, you see, and this is super interesting, that the TP53 single mutant has different expression profiles than the double mutant. So you can see here the red is a higher correlation with a protein expression marker, and blue means less correlation. So as you can see here, the TP53 single mutant seems to be monocytes with a high CD11b and high CD64 expression profiles, unlike the double mutant, which, the single mutant seems to be myeloblast with high CD34 and high CD117, and the double mutant here in orange seems to be monocytes. So this is the first time that at the, simultaneously having DNA and protein expression profiles, you can see the true clones that you could think they are the same population. I mean, they are different in genotype, but they might be similar in phenotype. They're actually different phenotypically as well. So the single mutant are myeloblast and the double mutants are monocytes. So super interesting to see. And they have done this with many more different populations. You can see as well the triple mutant clone and the expression profiles of different markers. Okay, perfect. So I will show you the next study, which is again genotype and phenotype, this type of myeloid malignancies. This is from Dr. Ross Levine at Memorial Sloan Kettering Cancer Center, and this has been published in Nature also last month. So last month we had the publication in Nature and in Nature Communications. So Dr. Ross Levine and the team are super interested in the evolution of acute myeloid leukemia and other myeloid malignancies, even pre-malignant states, such as clonal hematopoiesis. And they want to know, again, what mutations are acquired and the order of the mutations and the differences between severity of leukemias. And they also did DNA only and DNA plus protein. So this first slide is, again, a very powerful snapshot where you can see this is the oncoprint and the cohort of patients, the percentage of genes that were mutant for the different patients in the cohort. So you see the genes with a higher number of mutations were TET2, DNT3A, and RAS, etc. You can see as well, they analyzed different states, different disease states, clonal hematopoiesis, myelodysplastic syndrome, myeloproliferative disease, neoplasms, and then different types of AML. And again, and also the stages, if they were newly diagnosed, if they were refractory, if they relapsed, if they were chronic, etc. So a lot of information. You can see here that the number of mutations was higher as the disease of the severity of the disease, the severity of the disease increases. So the more severe the acute myeloid leukemia, the more number of clones you would expect. Also, the number of clones is higher as the severity of the disease also increases. When we look at the phylogeny of the different myeloid malignancies, you see here that for a clonal hematopoiesis sample, as this is a pre-malignant state, you have a big wild-type clone and then single mutant clones. So this is very interestingly, as we provide information about the zygosity, so whether the mutation is homozygous or heterozygous, and they actually had to rewrite their own bioinformatics pipeline in order to call homozygous clones, which is, for instance, this one is a single DNT3A mutation and a double DNT3A mutation, so it becomes homozygous. This is a clonal hematopoiesis sample, so again, pre-malignant, and the majority of the cells are wild type. Then you move to 2 different AML samples. Here they also describe that the mutations happen in epigenetic regulators first, such as DNT3A and IDH2 or IDH1, and then in signaling genes such as FLT3 or NRAS. So you can see as well, very interestingly, that you have here a DNT3A IDH2 double clone that acquires an NPM1 mutation, and you see the percentage is much larger than the others. So perhaps the acquisition of NPM1 is kind of providing a synergy and giving that clone an advantage to expand more. Same happens here. When IDH1 FLT3 acquires NPM1, now these mutations become, this clone become larger. So another thing they described was a FLT3 and an NRAS mutations being, what they saw was that they happened to be mutually exclusive. So either one clone has a FLT3 mutation and another clone has NRAS mutation. Right, so of course, they were interested in genotype plus phenotype. So then they used our DNA plus protein approach. And here as an example, you can see again that DNT3A and RAS double clones correlated with high expression of CD11B, whereas DNT3A FLT3 clones correlated with a low expression of CD11B. These are 2 clones that without the multi-omics approach, you could expect they come from the same cell population, but they are actually different cell populations based on expression profiles of membrane markers. And here you can see the same. So you can see different mutations that were defined. You can see DNT3A single clone. So orange is heterozygous, beige is wild type, and red is homozygous. And you can see the protein membrane markers that they assessed. Purple is high expression and green is low expression. So you can see here, for example, that the DNT3A FLT3 double clone is high on CD38, low in CD34, unlike the single DNT3A clone. So again, they seem to be different populations. You can also see the exclusivity of the NRAS mutation. So you either have one or the other or the other. You can see as well the homozygous and wild type clones for specific mutations. So again, very first time we see DNA plus protein for these myeloid malignancies and the correlation with the genotype. So for those who do flow cytometry or who are used to assess these expression profiles of membrane markers with flow cytometry, to finish off, I wanted to show you how well our data correlates with flow cytometry, which is the gold standard, right, for this sort of analysis. So this is the Multiomics DNA plus protein Tapestry platform. This is a mix of 5 different cell lines, and these are the 7 different markers that we assessed. So as you can see here, we highlighted 1 marker and 1 cell line to make it easier. And this is the correlation with flow cytometry. So you see the levels of protein expression correlate very nicely with what you could expect when you do the same analysis with flow cytometry. This is the exact cell line mix. So very nice to see the molecular biology on the left and the flow on the right. So as a summary and conclusions, the single cell or the Tapestry platform is the first and only platform that can detect somatic mutations, insertion deletions, copy number variations, and proteins simultaneously in single cells. It adds this simple staining protocol at the beginning of the workflow. It has been validated on cell lines and blood cancer samples, and essentially allows you to analyze genotypes to phenotypes. So with all that, I'd like to thank you for your attention and see if you have any questions.
Thank you very much, Gema, for nice and interesting presentation. We have time for one short, maybe 2 questions. May I ask a question? How many proteins can be analyzed simultaneously in Tapestry system?
Yeah, so right now we have launched our solutions with a panel from BioLegend, and it is 45 plex. So you could assess 45 proteins simultaneously, but you can even add more, you know, so you can add you can buy the 45 plex custom antibody from BioLegend, but if you want to add custom proteins to that, you could do, right? There is so far not a hard number of proteins that you could analyze, but we have validated so far up to 45.
Okay, thank you. And if you don't mind, I will ask another questions.
Sure.
About lazy microdissected samples. Is it possible to use these samples in Tapestry analysis?
Yeah, so I mean, as I said, so we can, it doesn't matter if you have laser microdissected tissue, as long as we can, if you have it frozen, well, you would have it fresh at that point, if we can extract the cells, right? So if the cell dissociation protocol is gentle enough, we can work with whole cells. If not, we could extract the nuclei of these sections.
Okay. Thank you very much.